Okay. I hope nobody has any objections if we record today's session. This event will comprise of a presentation by Jitka on Grasslands, the hidden part. You can see a brief introduction on the screen, and we will have a presentation which I guess will last something like an hour or a bit less. And after that, there'll be opportunity for discussion. So at that point, you can open your videos and microphones and discuss for as long as you have questions. And I will also record that if nobody has any objections, because that's often interesting for other people to see. I've also included on these slides mention of a forthcoming bigger event that we're working on together with DIDM, the Asian Grassland Conference, which is a virtual conference which is going to be organized in April. It's now open for registrations and for submission of abstracts until the 27th of February. And again, you can find more information of that via our website. But now it's 3:05 here in Finland, 2:05 in Central Europe, and I will pass over to Jitka to begin her presentation. So I'll stop sharing my screen and then you can share yours.
Thank you. Thank you, Stefan, for introduction. So I will start. Hopefully you see my presentation in presenting mode. Okay. So I will speak about belowground parts of herbs generally. So about these herbs in grasslands as well. And those, especially I will speak about roots, rhizomes, bulbs, and tubers, and so on. And I will speak about them from point of view of functional ecology. Small reminder for what functional ecology is. Functional ecology compare plants in order to find their strategies, to group them to the growth forms, for example, or some life history strategies. And with use of these groups to predict their response to environmental gradients. It is a rather old branch of ecology, so it started with ecology itself, because Humboldt was first author of classification of growth forms in plants. But functional ecology, this name is used only from late 90s, where were published two very influential papers in rather small journals, in Plant and Soil and Journal of Vegetation Science. So it means not in these big journals like Nature and Science and such journals. And these papers independently to each other established functional ecology as we know it now, formalized this approach to use some plant traits or plant groups for comparison and for studying plant strategies. In these two papers, author proposed that instead of using lifelong experience with behavior of plants, with flora and vegetation of the area, you can just go out and measure few easily measurable traits, which are important for plant function. And using this, you will get information about plant strategies. And why to use only few traits? Because anyway, these traits are coordinated or correlated together. So selecting proper ones, you get whole diversity of plant functions. And moreover, you will get not these categorical values like different growth forms, but you will get measured variables. And it is much more easy for calculations. And this attempt allowed like using students, sending students to exotic vegetation and let them measure leaves, plant height, and collect seeds. And with this information to build this new approach in functional ecology. In these papers, they proposed those traits and said that those are the relevant functions which are relevant to the traits. And in red, you have the smaller trait set, and it was proposed by Westoby. And in fact, his approach, right, really selecting 3 independent axes of strategy specialization was very inspirative because it was the most easy approach imaginable. And it led to compilation from the literature, measurement of new values in field, and accumulation of trait data into databases. And there was belief that the measuring these traits for as many plants as possible, it will help us to describe plant strategies and describe the response of plants to environmental gradients, to climatic change, and so on. After 15 years of effort, databases were filled so well that Diaz et al. Published first overview how these traits, these easily measurable traits, are correlated to each other, how they build these plant strategies. And they used plant height, stem-specific density, leaf area mass, leaf mass per leaf area, nitrogen content, and diaspore mass. And in this picture, we can call it like fried egg graph. You can see these points are species. The closer the points are, the more similar the species are in these measured traits. These arrows are pointing towards increasing values of these trait values. So you can see 2 basic axes in this picture. It is on this horizontal axis where it's going from small to large plants, and plants are accumulated in 2 places. Here, small plants, herbs, and large plants, trees. And second axis is going from plants with small leaves, thick leaves with low nitrogen content to plants with big leaves, thin leaves, and high nitrogen content. So acquisitive strategy versus conservative strategy. So it was like describing this knowledge for newly gathered on measurement, by measuring these traits, these plant strategies. Yes, that were like people saying it is nicely describing plant strategies. Even we like knew them before, so we knew that there are herbs and trees, but we have it measured now. On the other hand, those traits probably are not as good as we thought in prediction of response of plants to environmental gradients. So it was a little bit not happy. Well, people were thinking why we are not successful in this goal of functional trait ecology. So it's probably due to better description of environmental gradients, because we have like a standard protocol how to measure these plant traits, but we are measuring these environmental gradients differently in different parts of the world, so it is difficult to compare. It may be due to large intraspecific variability, and it may be due to we selected wrong traits, or not, that there are other traits which are important as well. And I will more speak about these wrong traits, whether there are some other traits which may be important. Which traits are accumulated in databases? So I will show it on this picture. So here is tree with realistic organ proportions. And now you can see a tree or plant with organ proportions reflecting trait sampling effort. So you can see that it has enormous number of leaves and flowers and fruits, but very tiny stem and nearly no roots. So effort invested the collection of traits to these organs was much lower than effort collecting leaf traits. And it's logical, because Westoby proposed to collect leaf traits and not root traits. He believed that root traits are coordinated with leaf traits, and as it is much more easy to measure leaf traits, so why to measure root traits? So we can see that from this list, only some of these traits were measured, or enough measured for big analysis. For example, this clonality and resprouting ability, dependent on below-ground organs, wasn't measured much. And moreover, as I already said, fine roots and nutrient acquisition was totally ignored. So the functional ecology must more focus on these below-ground parts, because they must maybe differentiation of functional trait strategies as well. And we don't know it properly. Of course, people studying roots, root biology, root ecophysiology, root traits, immediately start to be active. They were active before as well, of course, but they were inspired by these results, and they start to accumulate or already available data, measure new data, discuss which traits are more important than other traits on a root system. So here you can see the proposal which traits may be measured on roots. It's enormous number of different traits, and they are usually rather difficult to measure, at least in comparison with leaf traits, of course, because at least they are hidden in the soil. After a few years, it was now much faster. Root ecologists accumulated and cleaned the data for few, of course, few easily measurable root traits, fine root traits. And this is nitrogen content, root diameter, root tissue density, a specific root length. And they did their fried egg graph, which is very similar to this previous one, except it has only one egg in it. And horizontal axis is showing a negative correlation between specific root length and root diameter. And it is interpreted as do-it-yourself strategy here. Either plant has fine roots which are searching for nutrients, or cooperate with mycorrhiza. So the plant has roots with high diameter that use mycorrhiza for getting nutrients. Second axis is similarly as in Aboveground traits, from acquisitive strategy, high nitrogen content in fine roots, to high root tissue density, like conservative roots, which are long-lived and so on. So from conservative to acquisitive strategy. Interesting is that these strategies are not correlated with this above-ground strategy. So they are really independent from above-ground plant strategies. Still, we are missing something below ground. This was done for finer roots, but we have coarse below ground organs. They have storage function. They provide plants with clonal growth. Why they are overlooked when we are already working below ground? There is a lot of people now starting to collect fine root traits, and still they are missing these coarse organs. I think it is due to, like, you are so focused on your focal plant organ, as it is shown in this picture, that you collect just stem, leaves, and fine roots, measure them, calculate, and then you create picture of a plant according to these measurements. And this may be much different from the reality. It's very funny when you look at different graphical abstracts in or illustration, conceptual illustration in papers dealing with some below ground traits, below ground ecology, soil biology in connection with plants. And when you look how these plants in these papers are depicted, it ignores that plants have roots, thick roots, or that they have rhizomes or bulbs or anything. This drawing, this green brown drawing here, I took from the paper describing steppe vegetation. So as researchers on dry grasslands, you probably recognize that it's not typical steppe vegetation. It looks like flower beds. So the people are so focused on what they should study that they don't see plant as a whole. I was thinking probably it is because these coarse organs, these rhizome bulbs and so on are only tiny fraction of plant biomass, so we can easily overlook them. So we try to find literature data about whole plant biomass in herbs and how much plant is investing to rhizomes. So what is biomass of rhizomes? And it is here, and you can see that it is in average 30% of whole plant biomass, a little bit more for eudicot plants, and in monocots, there is big scatter of points. So it is from 5% to 80%, but it very much depends on data. So these data about whole plant biomass are usually from cultivation. So it depends on age of plants as well. So we need better data, of course, but at least we know that it is really substantial part of a plant, so we cannot ignore it. And it's not only biomass, it's diversity of forms and traits. It's not so simple to say those are below ground stems. Those are different organs are participating on this coarse organ. So it may be thick root, it may be stem, it may be bulb, which is composed from storage leaves, and it may be a root from which plant may regenerate. All this diversity may be classified to, let's say, below ground growth forms or clonal growth organs, as we did in CLOPLA database. So CLOPLA database I started to work on it in 1993, so much earlier than these papers about functional traits. But like online version of the database was published in 2006. And CLOPLA database is part of Try database, and its aggregated form is freely available as data paper in ecology. So the data are available about clonal growth organ or belowground storage and clonal growth organ of Central European flora. But so far it is only flora which has its database of clonal growth organs. The most common in Central Europe are the following types of belowground organs. It may be annual plants, so without perennating organ belowground, or non-clonal perennial that have perennating belowground parts with bud bank but cannot grow clonally. And then stolons, different types of rhizomes, or bud-bearing roots. Of course, we have in Central Europe bulbous plants, tuberous plants, but they are not so common. We know from analysis of Central European flora that these organs are in phylogeny, they are as a flexible transitions from non-clonal plants to clonal plants. And we have two types of clonality which are independent. And this is clonality based on bud-bearing roots, on adventitious sprouting from roots. From non-clonal plants, it may develop or go back rather easily, or it may go from non-clonal plants to plants with epizootic rhizome, and then other types of rhizomes or stolon or back to non-clonal plants. But these stem-derived types of clonality are separated for root-derived type of clonality. Still, we are speaking about functional ecology, so these organs has their traits which may be measured across these organs. Not so in all type of these organs you may measure bud bank size or bud bank depth, or different clonal traits like lateral spread, multiplication rate, or persistence of connection between ramets. Or you may measure a storage size and type, or woodiness of these belowground structures. And these morphologies, these stolon, rhizomes, bud-bearing roots, are to a certain degree different in these traits, but sometimes they are similar. For example, persistence of connection between ramets is very low for stolons, but very similar among rhizomes and bud-bearing roots. On the other hand, offspring number is highest in stoloniferous plants and lower in all other categories, or spreading distance by lateral spread is high in the stoloniferous plants and bud-bearing roots and lower in rhizomes. Bud bank depth, of course, is very low in stoloniferous plants, where the stolons are above ground, whereas it's deep in epizoogenous rhizomes and hypogoogenous rhizomes, and bud bank size is higher in bud-bearing organs and rhizomes, whereas it is low in epizoogenous rhizomes and non-clonal plants. But we can measure these traits across these organs. And what are the functions these clonal traits or I am using clonal because like originally I studied these below ground organs to understand clonality, but always when you want to understand clonality, you must compare with non-clonality. So it is included, of course. So of course, function is like clonality. It is vegetative regeneration. It's demographic function. So plant may reproduce not only sexually, but vegetatively as well. But it plays a role in nutrient acquisition as well. Yes, a non-clonal plant may forage by roots, but clonal plants may, as it grows horizontally, it may forage by these horizontal structures like rhizomes, stolons, or horizontal roots as well, or response to disturbance. So they may be seeder, regenerating only from seeds, but they are sprouters, they rely on bud bank. Of course, these organs play a role in response to stress, but we know only little about this, and they surely play a role in effect on ecosystems. And I will now show you examples from each of these functions, what we know. Sometimes it's random selected examples and more of them exist. Sometimes it's only one thing we know about whole thing. So I will show you something. So the first, clonality. It's traditional hypothesis existing about clonality is that it differs among communities and is most important in conditions where sexual reproduction is difficult, where it's stressful conditions. So I will show you how clonality is distributed in European habitats. They are here listed according the number or percentage of clonal plants. So you can see nearly 100% of clonal plants are in standing waters, and other very wet communities, poor fens and mires, running waters, fens, wet grassland, bogs. So those are communities with high percentage of clonal plants. Whereas these ruderal communities, which are often disturbed, they have low number of clonal plants. So I don't know whether it is like stressful, whether we can interpret it so easily that the clonality is prevailing in stressful environment, because standing waters, like yes, they may be stressful because it's water and it's limiting plant growth, which are not adapted. But there are plants which are adapted to standing water, and it is an environment for them. I would say the clonality is easy there because plant may easily fragment, they may easily take new roots. So the clonality is easy. I use these marks like to show you differences between different clonal traits for these few communities, just to highlight them. So you can see the highest number is in water, lowest number of clonal plants is in arable land. In alpine grasslands are here with a lot of clonal plants. It's stressful environment, but and we have this grasslands, wet grassland, messy grassland, and dry grassland. So this gradient is more from wet places to disturbed places. What about other clonal traits? Now we have lateral spread, and it is only for clonal plants now, how good they are in lateral spread. So again, standing waters, they have the highest lateral spread because they have a lot of these plants which may be freely moved by water, that have different turions or small fronds, which plant may be split into few parts, and they are distributed by water currents. Arable lands, which had very a low number of clonal plants. These clonal plants are characterized by high lateral spread. And we can see even dry grasslands, which have less clonal plants than wet grasslands, have higher lateral spread than wet grassland. And alpine grasslands, which had like very high number of clonal plants, all of them has very limited lateral spread. So now you see that something different is when you characterize your community about according the percentage of clonal plants or according the lateral spread. And another trait is multiplication rate, how many new ramets one ramet is producing per year. And again, you can see that standing waters are highest number of highest value of this multiplication rate. The second are arable land, again this community where there is a low number of clonal plants, and again this dried grassland, wet and mesic grasslands, and alpine grassland have very low numbers. Example with nutrient acquisition. So yeah, we know that these organs do not participate in nutrient acquisition. They are not fine roots. They are not leaves that acquire carbon, but they connect these green above ground parts with fine roots. And as they sometimes grow vertically, sometimes horizontally, they probably would affect nutrient acquisition. So back to this graph, which I already shown, these root traits. And we tried to look whether these root traits are connected somewhat with this coarse organ, these types of clonal growth organ or non-clonality. So here you can see I shown our results in this original graph by Carmona. So you can see that in this acquisitive gradient, it's really this nutrient-rich fine roots in places with high nitrogen, high disturbance frequency, and high moisture. They are showing up. So those are like very, very good conditions for plant growth. And these colorful points are centroids of these clonal growth organs. So these are annual plants, stoloniferous, both types of rhizomatous plants, non-clonal perennials, and root-sprouting plants. So this gradient is showing short lifespan of rooting unit because annual plants are just annual. Stoloniferous, usually these ramets are very short-lived. But in rhizomatous plants, they are very universal, occurring everywhere. So they don't have such pronounced traits. But these non-clonal plants are more this conservative part, and their rooting units is long-lived, similarly as in these root sprouters. So you can see that while these fine root traits are not connected with above ground traits like leaf traits or seed traits, they are a little bit connected with these coarse below ground organs. Another example is from sprouting. So we know that below ground coarse organs store carbon and buds for regeneration of plants after recurrent disturbance. And it's question whether how they differ along disturbance gradients. For this, we had to analyze disturbances in Central Europe. So we prepared something like Ellenberg indicator values for disturbance severity and disturbance frequency. And it was done on the base of expert knowledge how individual plant communities are disturbed, how often, frequency, and how severe. So it means how big places or patches of the communities are disturbed, and whether the disturbance is even affecting soil surface. So it is disturbance severity. So somewhere here is disturbance 1 per year, like traditional meadows. Those are forests. So you can see here a distribution of woody species in this space of disturbance frequency and severity. And here you can see annual plants. And we use this indices for individual species to analyze plant traits. What we have here, so this is disturbance frequency as return time, so it is once per year, once severe disturbance once per 100 year, and disturbance frequency. Here you can expect something like arable land, and here you can expect something like forest. So you can see that along disturbance frequency, the annual plants are increasing in frequency in these places, and clonal plants are decreasing. But there is peak of bud bank and little bit also in the clonal plants when disturbance return time is about 10 years. So the plants have a larger bud bank. And a lateral spread of clonal plants is following this pattern. According disturbance severity gradient, you can see again annual plants increasing, clonal plants decreasing, bud bank is decreasing from the beginning, but lateral spread is increasing. So it is the same result I have shown for arable land, where it's very large lateral spread. So now about the response to stress. We started thinking about response to stress and these cores, belowground organ, only recently, because I was always interested about clonal growth, and I believe that it is main function of these organs. But when you are thinking about this, of course, those are storage organs as well. And when plants build a storage of carbon, it is for future use, for the use when there is scarcity of resources or when there is disturbance. There are like two hypotheses which are going against each other. As the belowground storage organs mitigate the stress, so plant is accumulating this storage to be able to respond to stress and disturbance, or storage carbon accumulated in stressful conditions because plants cannot grow, so it is accumulating surplus of carbon, even it is not using it. Of course, it is not easy to quantify carbon storage for numerous species, because analysis of carbohydrates is very laborious and expensive analysis, and we don't have data for a lot of species. But we used drawings from ClopPla database to measure volume of storage organs. We selected rhizomes only, and only those where the annual increments are clearly distinguishable as in this rhizome. You can see here 1, 2, 3, 4, 5 years old rhizome. We measured length of lateral spread, diameter, and number of years for which the rhizomes is persisting for these epigeogenous rhizomes and hypogeogenous rhizomes. And we calculated volume from these measures. And you can see how these measures contribute to volume of the rhizome. So this is a rhizome total volume, and the main effect on this has diameter of rhizome. So when you want to have voluminous rhizome, it's better to have a thick rhizome than to have long rhizome, because lateral spread and persistence are contributing, of course, as well, but much less, and they are negatively correlated to each other. So when plant has big lateral spread, it has usually low persistence. And now, what is a relationship of these traits to environmental gradients? So when we take total volume and gradients of disturbance severity, disturbance frequency, nutrients, moisture, and plant height. Nutrients and moisture are Allenberg indicator values. This disturbance severity and frequency are these indices as I described above, and plant height is like very much affecting the total volume of the rhizome. So you can see that with increasing disturbance severity, the volume is increasing. So you can interpret it like plant needs big storage because there is severe disturbance which is coming often. So plant may sprout, but when the disturbance is coming very frequently, the total volume is lower. Nutrients are not affecting total volume, moisture is affecting positively. And there is big effect of plant height. When we do the analysis where the plant height is filtered out, it is here, we can see that all significant relationships disappear, and all these values are shifted to bigger volume. So in the same plant height, all these disturbed and very productive habitats tend to have larger volume of rhizomes than one would expect. Here I have the individual analysis of the same type for these measures. So it was total volume here. And when we take only lateral spread, we can see that lateral spread is responsible for this effect of disturbance severity. Again, this is this effect on arable land perennial weeds, which have big lateral spread, and effect of moisture, which is supporting big lateral spread as well. Diameter is responsible for this effect of height, and persistence of rhizome is responsible for lower volume and frequent disturbance. So when you have frequent disturbance, the rhizomes will be probably very short-lived. Now, let me speak about effect on ecosystems. It's even, I said that connection of this belowground clonal organ and storage organ, the stress is something what is not very much studied. And but the effect on ecosystems, I think, is not even, it is even less studied. Even I think it must be very important for carbon sequestration. As I said, the 30% of plant biomass may be in rhizomes. Protection against erosion, because this rhizome net may keep the soil on the site. And of course, buffering biomass production due to with the help of bud bank. In which ecosystems this may be important, these functions of below ground organs. It will be probably in ecosystem which has big below ground biomass. Here you have the main biomes on the earth, and these brownish ones are those which have more biomass below ground than above ground. And it may be 5 times more, 20 times more biomass below ground than above ground. And this biomass below ground is not only finer roots, of course, those are coarse roots, rhizomes, lignotubers, and so on. And we can expect that these organs affect really the functioning of these ecosystems. I will show you only some examples which may be useful for dry grasslands. I selected 2 examples from dry grasslands. One is from in Mongolia, and one is from American plains. And I will present them on background on the new conceptual scheme of aridity and tipping points by Berdugo and Maestre. So they are saying that with increasing aridity, there is not continuous change in ecosystem functions, but there are thresholds where everything is changing abruptly. And they are describing these thresholds like along aridity 0.5 or 0.7. So here vegetation is declining, the cover of vegetation is decreasing. Here is this soil disruption phase. The soil cycles of nutrients are decoupled from plants, and soil biological activity is decreasing, and there is breakdown phase when even higher aridity. And they are proposing what is changing, especially in traits of plants, so productivity of plants, leaf traits, and content of nutrients. But not surprisingly, they are not looking on below ground plant traits. But we are not without data or without published data about what is happening about this aridity gradient. And I will show you 2 examples. So these American data are from plains, from precipitation gradient, from Dalglish and Hartnett. It was published 2006 in New Phytologist, and you see these few places from different prairies. There is long-term annual precipitation and average bud bank per 1 square meter. And you can see that it is decreasing towards a dry condition. And what does it mean? So here in this moisture part, the prairies are dominated by perennial plants, rhizomatous and non-rhizomatous plants, both. But when we are going to drier conditions, the cover of perennial plants is decreasing and annual plants are taking over. And they are relying on seed bank, not on bud bank. Perennial plants are relying on these rhizomes, tubers, and root crowns to regenerate each spring. And but these annual plants are relying on seed bank. Seed bank may be very rich in seeds, but it is dependent on special triggers to start to germinate. And these triggers, environmental cues, may not come each year. So the variability in biomass production on this end of this aridity gradient is very high, whereas here is much, much lower. Example from Inner Mongolia, these points are sampling points. It is going from wet regions with 500 millimeters precipitation to Gobi Desert with less than 100 millimeters. And you can see here along this gradient, a vegetation cover of all plants, as you would expect on this aridity gradient, is decreasing, relative richness of annuals is increasing, biomass proportion of annuals is increasing. And bud density is from the beginning increasing a little bit and then abruptly decreasing to very low numbers. So the same effect. So these arid places have not this bud bank which buffer biomass production. And here we have, we can visualize this tipping point. And we have even more detailed data for this steppe vegetation in Inner Mongolia. So prevailing buds in bud bank are on the base of tillers. So it means tussock grasses, for example. They are nearly everywhere equally represented. But these rhizome buds are decreasing very much, and in these arid conditions, you will not find them. So it means there are no rhizome, no horizontal structures to keep the soil. And it's not only that these rhizomes are missing, but plants here in these arid conditions don't need to search for water and nutrients in horizontal direction, but in vertical direction. So they prefer probably to have deep roots, not to have a big lateral spread. But it means that they have lower bud bank for regeneration, seasonal regeneration or regeneration after disturbance. These bulbobuds, so it means bulbous species which are typical for dry steppe vegetation. You can see that they are not affected, they are even supported in these arid conditions, and there are buds on dicotyledonous plants as well, and they are nearly missing in these arid parts. So we can say that behind this decreasing biomass along this aridity gradient, there is not only leaf traits, changing leaf traits or changing abundance or clumpiness of vegetation, but it's especially this bud bank and rhizomes. Those are missing, and increasing of annuals that are dependent on seed banks. So, and it may be very important part of this aridity in tipping points, which wasn't so far considered. Because we don't have such arid places in our Central European flora where I am working, I will show you briefly some results from Our Grasslands. It is Aboveground biomass of grassland and rhizome biomass on the level of community. So it is not for individual species, it is per 1 square meter. So you can see how its rhizome biomass is increasing with whole biomass of grassland. But when we look at at the details, we can see that these data were collected in two types of grasslands. One type was mown once per year, or was less productive, not capable to produce so much above ground biomass to be harvested 2 times, but it has a higher biomass peak than this intensively used grassland. So this less intensively used grassland have really a large amount of rhizome biomass, but this more intensively used grassland have lower amount of rhizome biomass. So this aridity may be, this effect of aridity may be multiplied by management of this grassland as well. As we can see in this study, which was short-lived, short-term study in American prairies, in Konza Prairie, in this part of the graph, you can see ambient precipitation. There is limited water accessibility, drought treatment, and this dark parts of graphs are showing grazed plots and this light ungrazed. And this is for grass buds. So bud bank, but one grass shoot. They are affected not by precipitation, but by grazing. When it's ungrazed, so they have more buds. In sedges, there is effect of drought, but not effect of precipitation. And in 4 buds, or there is some interaction, and in 4 buds, there is strong effect of drought. So you can see that the effect of drought versus effect of management may differ between groups of plants. And of course, it may be a reason of degradation of grassland, of dry grassland. For example, this is a picture from China, where it's a restoration effort to protect or to restore degraded grassland in Mongolia. So to conclude, yeah, these coarse below ground organs have different functions. We know them only. We know only some of them and not properly, so there is a lot to do and a lot to study. And of course, to study these below ground coarse organs, rhizomes, tubers, and bulbs depend on knowledge. So we are trying to teach people to recognize these organs and to study them. We publish a handbook of standardized protocols for collecting these traits. We are organizing course Go Below Ground, where you can try measurements of these below ground traits. And that is all. Thank you for your attention, and thank all my collaborators for contributing to this lecture.
Hi, Jitka. That was a very interesting talk. Thank you very much. And personally, I have many questions, but yeah, of course. Now it's time for questions. So in the audience, if you would like to ask questions, you can raise hand and just speak. Or if you want, you can write your question down to the chat and I can read for you.
Please ask questions, or otherwise I will ask you. For example, are you considering to start to measure below ground traits in your plots?
Okay, then I will start with my question. So you can consider me as a kind of ecologist, grassland ecologist interested in vegetation, but yeah, of course, with your presentation now, I am more interested in below ground organs. From a very practical point of view, would you say that, okay, you are just going and measuring above ground stuff, but not to miss major information, it would be good to focus on at least 1 or 2 or 3 below ground traits. Would you say them?
Yeah, we were thinking about this because, of course, like to go to species level, to measure these traits is not easy, and it needs some morphological knowledge, or yeah, it's, of course, it's a lot of work. Whereas all of us may recognize leaf, it's not problem, but below ground, yes, you may recognize fine roots from other things which belong to plants, and this may be like way to start, but it's very laborious, for example, to get below ground biomass. On the other hand, you don't need any special knowledge for this. So to know what part of biomass in your community is below ground, and which part of this below ground biomass is fine roots versus rhizomes, bulbs, tubers, may be interesting, but it's a lot of work. On the other hand, I would be satisfied with this biomass of these coarse organs, like thicker than 2 millimeters, and let all these fine roots, because with fine roots, you must go sometimes really deep, and it is not always easy, especially in grasslands. Whereas these coarse organs are usually concentrated on in upper soil levels. So biomass might be one of the things, but when you are doing it on community level, like to measure some traits like below ground dry matter content for individual species of these coarse organs, maybe like easy things to do, like to dig up species by species from, again, these coarse below ground organs, which are not so deep, to weight, fresh weight, dry it, and to weight, dry weight, and you have dry matter content. It may reflect like something like SLA, of course. Yes, either you invest hardiness of these organs, they are usually in this, when you want to have them long-lived, or you have short-lived organs, so you have bigger turnover, for example, because you have enough moisture, enough nutrients, so you have low values of dry matter content. Those are easy traits.
It sounds doable, and as a start. Yeah, Steve.
Yeah, thanks very much, Jitka. It was a very broad-ranging presentation with a huge amount of information. One thing that I wondered about, you were looking a bit at disturbance, and you were looking at precipitation and factors like that. I was wondering, do you see any... Oh, now my phone is disturbing me. One of the things with climate change is we expect increasing intensity of extreme climate situations. I wonder if we would be able to see in root traits that maybe some of the habitats you were looking at would maybe have different levels of resilience to climate change.
I think especially this dry end of grasslands is worth the study in this respect, because when I visited like in Mongolia, so you can see these degraded grasslands, and it's very interesting because it's covered by annual vegetation totally, because they are not in places which are so dry as Gobi with shrubs and so on, but there are big annual herbs which die out in autumn, and in winter it's not covered by anything, this soil. And there is big soil erosion, terrible soil erosion, like it was in 30s in American plains, and it's very difficult to reverse this situation, like to plant something because it became dry and this soil degrade. So I think this tipping point or this end of grassland on dry part, even we probably don't think here in Central Europe that we have these problems. I am worried a little bit about our grasslands, dry grasslands, which do not look like end of close to desert, but still like few years of drought and maybe overuse, like grazing, or probably not mowing, but grazing may degrade it. So, but I am not sure in wetter end, and I am not sure, for example, in grasslands which are dependent on soil, on snow cover in mountains, this may happen something as well, because these plants are adapted to be covered in winter by insulation layer, so they don't have freezing temperature. But when they lost, and we observe it in Central Europe in mountains, where we used to have 3-4 months of snow cover, we have 1 month and it's thawing and again snowing and so on. So this, I think, will change these communities as well.
Thanks. I just read, there's one question in the chat, which I think is, maybe by mistake, has only come to me. My colleague from here in Finland, Päivi Virnes, wrote that here in Finland, Salicornia pernans, which was formerly Salicornia europea, is strongly dependent on extreme drought and high disturbance. Do you know anything about the ecology of that species?
No.
Okay.
Sorry. But I think it is not rhizomatous. No, I don't know, simply.
Yeah. Did you want to comment, Päivi?
Then I can ask my second question. So I think you showed us a graph with disturbance severity and frequency. So frequency is kind of easy to measure, how many times in a year or how many times in 100 years. But how do you measure the severity when you consider forests, grasslands, and everything?
Yeah. It was done by Milan Chytrý and his experienced guess. He took plant community types, and each occasion he thought about how often it is severely disturbed. Of course, everything nearly is disturbed, like slightly, but what is disturbed severely? And whether the disturbance is affecting one plant or patch or whole community, or even affecting soil. in such categories. It is described in a paper from 2016 in Journal of Vegetation Science. And then this index is calculated according to the occurrence of the plants across the communities where they have the optimum. It is weighted by the optimum and the occurrence in all possible communities.
I mean, for me, like a clear-cut forest management would be the kind of both frequent. It may be both frequent and very severe because it removes all the vegetation. But now I guess in Central Europe, there is no clear cut, so it is not. I mean, the graph you show, forests were kind of in the less severe part. So I was questioning why forests are not at the extreme part of severity.
Yeah, because like for herbaceous vegetation, it may, of course, it depends how it is done. So it may destroy everything, but at least for herbaceous vegetation, it may not be so catastrophic as for trees. So it is not so severe as arable land when you have 2 times per year, like shifted the soil and upside down.
Yeah, okay.
And it is, of course, partly correlated because you can, when you have severe, the most severe, usually the most common the disturbance is. And it is also a little bit correlated with nitrogen availability, because these arable land are typically, so these plants can survive only because there is enough of nutrients. So there is also like some correlation with Allenberg indicator values for nutrients.
So Jitka, I also have a question. Thank you for this interesting talk.
Thanks.
You said, well, you made very clear that below ground rates are important and you called that we should measure them, but well, as in above ground rates, many people use rates, but they don't have the time to measure them, but they, well, make the shortcut to go to databases, download them, and try to do something meaningful with it. So my question is, how good is the coverage of the relevant below ground traits nowadays in databases? Does this make sense to calculate community weighted means for?
It depends where are you working. If you are working
let's say in
Central
Europe, so how big is the coverage for the Central European flora for some key?
I cannot say 100%, but it's close to, because the CLOPLA database contain whole flora of the Czech Republic. Of course, they are not 100%, but they are nearly all plants. So for
which traits? So CLOPLA does have all, no, certainly not all the below ground traits, but some key traits.
Of course, it has these traits which I described for this clonality. So it has type of clonal growth organ, whether it's rhizomes, stolon, etc. It has lateral spread, multiplication rate, persistence of connection between ramets for clonal plants, not for non-clonal, because it has no sense, but you may recognize whether the plant is clonal or non-clonal. We have aggregated version, we have non-aggregated version where are all available information from literature and from our own studies. It is online as CLOPLA database, or it is included in TRI. But there is aggregated version where you have only one value, averaged value for species. So you may easily use it per species level and not think how to aggregate this data. And this is published in Ecology, and it is freely available as well.
Thank you.
You're welcome. And there are papers where we are explaining these traits and their functionality. So you may consult them which traits may be important for your question. It would be nice to have like more data, of course, from other grasslands. So I worked in Mongolia, but the data are with my Chinese colleagues. So it's a little bit, we are not finished yet, but these are not available. And we are working on American grasslands. We have limited number of species because I studied Central European plants for 20 years, but I studied American plants for 1 year. So of course it's limited number, and due to pandemic, we were not able to continue with our work. So we hope in future we will be able, so American prairies will have, because they are still similar to Central Europe, to European flora, so we are able to describe them. Of course, for tropical grasslands, there are data, but they are not standardized the same way, so there are different categories, and data are scattered in literature, so there is no database available.
Thank you.
I had one other question still, Jitka. I was thinking about carbon sequestration. There's, of course, a lot of interest now in studying carbon sequestration in many vegetation types. Would you say that there may be benefits from looking at traits that are the traits that would be correlated with carbon sequestration, that we could use particular trait groups to help get a handle on carbon sequestration?
I am sure that this key for carbon sequestration in soil is this existence, biomass, and turnover of these coarse below ground organs. What is studied is fine roots, biomass, turnover, and exudation. But these coarse organs are not studied. What is studied is rhizodeposition. So when the fine roots die out, what happened with them in soil. But we don't know what happened with rhizomes when they die. Because what is interesting on clonal plants, not only that they are growing clonally, multiplying, so it is interesting enough, but why they are doing it? Because they are dying from old parts and growing these new parts. And what does it mean, dying from old parts? It means that these rhizomes are decaying and staying in soil, and all this carbon, maybe this carbohydrate storage, maybe storage in this rhizome remains or is used for this new growth. We don't know. But it must be important part of carbon input into the soil. But we don't know much about this.
Okay, thanks. So anybody else have any questions still?
No questions, but just a note. Hello, Stephen.
Hi, baby.
Can you hear me?
Good to see you here.
I'm still here. Have you worked together with a Meyer or Pog or Marsh? I don't know what's the correct word of ecologist, because the carbon storage is a really a big question in these habitats. And here in Northern Europe, many of the traditional hay meadows are actually also, I'm not sure if you're familiar with the word Arbemeyer. Stephen can tell you it's the maybe the only word that has been originating from the Finnish language, the scientific language, Stephen knows what's Abermeier, don't you?
Yes, a kind of peatland that's typical of the northernmost.
Yeah. A flood influenced, a spring flood influenced mire, which has been traditionally cut for hay production. And the most fertile and the most hay producing Abermeyers, called as letto, have are also traditional cultural biotopes. You should, I think, cooperate with the Maya conservation people, because you have much in common.
Yeah, I think where the rhizomes are not denied to play the role is in seagrasses, for example, when we are in wet place. I know that you mentioned a little bit different things, but because it is acknowledged that rhizomes are important there for functioning. And in wetlands, generally, people know, because the biomass of rhizomes in wetlands, due to big productivity, as I said, it is correlated with aboveground biomass. And when the wetlands are productive and not used for cutting, so they have really big biomass below ground and above ground, and they have big amount of rhizomes. And I think these people know it. Yeah, but when you are going to the dry end of the gradient, or you are generally botanists know that plants have rhizome. It's not problem. Everybody will agree with it, but it doesn't prevent them to ignore them in their studies. I think they would say, if we study clonality, we will surely include it, but we are studying something different.
Sorry, I'm a conservation biologist. I was looking at this topic from the point of the conservation of a rare species, as Stephen well knows. and I was thinking also the carbon sequestration, but also the conservation of the biodiversity, how to maximize the surveillance of the very rarest species we have on wetlands and flood influenced areas, for example.
It's complicated topic. I think I cannot give any simple.
Sorry, Stephen.
I think
you will regret you invited me here.
Certainly not. I think at this stage I'm going to switch off the recording. So if there's anyone that's shy to ask a question while the recording is switched on,
please do.
and then we can switch to a more